Okay, could somebody confirm you can hear me?
Yes.
Yeah, we can.
Thank you. Whoop. I think somebody dropped. I don't think you can see my screen, so let me show it. Actually, before I show it, let me state that I have done some grading. I'm only halfway through grading Lab 3 for the first time, and I hope to finish Lab 3 for everyone by tonight so that you can turn it in for full credit this Saturday. Lab 4, I'm not going to get graded anytime soon. So its due date will probably be later than this Saturday. Any question about any of that? I'm talking about the grading of Lab 4, the second grading. Any questions about any of that? Let me go to I think I got the lecture. The last lecture graded as well. And maybe some extra credit study, your group study. You got the pictures graded. Anyways, hopefully I will get Lab 3 done tonight, and then it will be due this Saturday at 11:59 p.m. for the second grading. Any questions about anything before we begin? All right, let me get out of the grade book. And then I'll show my screen. All right, you should be seeing my screen now. Today is January 30th, and we're going to be talking about Lab 5, Streaking for Isolation in Culture Media. You should also read the unknown project instructions file on Canvas for next week. Next week, I'm going to discuss the unknown project, and you will begin it. So you need to read the instructions. Let me show you where that is. I think it's week 5 lab. Oh, I need to get that showing. So in week 5 lab, the first part is the unknown report. The unknown instructions.rtf is the document I want you to read. I'll talk about it next week. Any questions? Let me remind you, if you have not picked a infectious disease topic, you need to clear it with me before you begin, because everybody's going to have a different project. And I also want to make sure it's an infectious disease and that you're narrow enough. A lot of people have done a very broad topic, like somebody did E. coli infection, and I said, no, E. coli causes a number of diseases. Pick one disease caused by E. coli and then write on that disease. And then other people wanted to do pneumonia. And I said, no, there's many different agents that cause pneumonia. So pick one agent that causes pneumonia, and then you can write your report on that. But you need to check it with me now, because a lot of projects have now been taken. And so when you ask for a project now, you should ask for at least 2 in case the first one has been chosen. I've even had one student who asked for 2 projects, and I had to get back to her and state that, sorry, both of those projects are taken. So do get started on your infectious disease project. That project is going to be coming due fairly soon. Let me look on the syllabus when that's due. It's going to be due Saturday, February 24th. So in about 3 weeks. Any questions about any of that? All right. Oops. I'm trying to move that up and it's not moving. Let's go on to lab 5. Lab 5, cultivation of bacteria, culture media, inoculation, and isolation. There is some reading, as there is for every lab from the textbook. Not too much today. A few pages on reading about culture media, and then a few pages on, actually, really only 1 page, maybe 1.5 pages, on obtaining a pure culture. Into today's lab, we will have some video clips for you to watch. The learning objectives for today's lab: 1, be able to understand the importance of proper specimen collection and transport to the microbiology lab. 2, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between the various types of culture media. 4, explain the purpose of streak plate culture technique, aka streaking for colony isolation. 5, define the following terms: selective media, differential media, and reducing media. Any questions on what we're going to cover? All right. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they'll dry out, and some are sensitive to exposure to oxygen, most anaerobes, for example. And these require a special transport medium to keep the specimen alive. Once a specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps to process it. First, there's 1, inoculation; 2, isolation; 3, incubation; 4, inspection; and 5, identification. In inoculation, you're just placing the specimen on an artificial culture medium to initiate its growth. Isolation is spatially separating the microbial growth so that you get distinct colonies. Incubation is placing the culture medium in an environment with conditions to facilitate the growth of the bacteria or the other microbe. Inspection is examining the culture for the presence of microbial growth and beginning to characterize the specimen. Identification is performing specific tests to ID any pathogenic microbes that are isolated. The 5 steps will be described as we're discussing different types of culture media. So the first type of culture media you're already familiar with, and that is general purpose media, such as Na or nutrient agar, Na or nutrient agar with yeast extract, and TSA or tryptic soy agar. These media grow a large number of organisms. They supply the nutrients for many things to grow. Not everything will grow on them, particularly fastidious organisms, will not. And these media are not selective. To promote the growth of fastidious microbes, these are microbes that require specific nutritional requirements before they can grow. We use an enriched general purpose media. For example, you can put 5% sheep's red blood cells into a tryptic soy agar, a general purpose media, and then you get an enriched media, also called an enriched general purpose media. One organism that requires an enriched general purpose media to grow is Streptococcus pyogenes, the bacteria that causes strep throat. While I'm talking about fastidious microbes, I can tell you that just recently, in the last couple of years, I'm not sure when it was first discovered, but I just read about it. I think it was in the last week that they finally figured out what they need to add to tissue culture, what nutrients to get syphilis to grow in tissue culture. This is an organism that they've been trying to grow for over 100 years, and they could never figure out what they needed to add to the media to get the bacteria to grow. But recently, the last few years, they finally figured it out, and syphilis can now be grown in tissue culture. Any questions about any of that? All right. There are also selective media, and these media contain chemicals that inhibit or halt the growth of some organisms, but they allow other organisms to grow. Why they're called selective. You should keep two things in mind when using selective media. First, a selective media isn't necessarily perfectly selective, meaning if given enough time, organisms that are selected against can grow in the selective media. Just that they grow slower. Second, even those organisms that are supposed to grow may form smaller colonies and may grow slightly slower than they would on a general purpose media. That's selective media. Let's talk about differential media. Differential media contains chemicals that produce visible differences among different groups of organisms when grown on that medium. These differences are unrelated to how well the organism grows on the medium. They just will make the two cells appear differently. Usually, they'll have a different color, but they could be distinguished from each other in other features as well. Lastly, we have reducing media. These media promote the growth of anaerobic bacteria because low or no oxygen conditions are required for many anaerobes to grow. Any questions about the types of media? All right. It's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. Let me show you the picture of that. There we are here. McConkey's agar is selective in that if you see growth on the McConkey's plate, then it's likely to be gram-negative because gram-negatives will grow on McConkey's agar, or MAC is a shorthand. And gram-positives tend not to grow. They have, you don't need to know this, but they do have bile salts in it. And what else do they have in it? Bristol violet, which gets in the cell wall of gram-positive cells and then keeps the cells from growing. Like I said, you don't need to know exactly what's in McConkey's, just that it's selective and it allows gram-negatives to grow. Gram-positives tend not to grow. So if there's growth on McConkey's, then it is gram-negative. And McConkey's can also be differential, differentiating the cells on their ability to ferment the sugar lactose. If the bacteria can ferment the sugar lactose, the bacteria will grow pinkish on the McConkey's agar. The sugar lactose will be fermented, creating acids, which will lower the pH, which will change the color to pink. And this bacteria is a lactose fermenter. This bacteria is gram-negative because it's growing, but it's not a lactose fermenter because the bacteria are not growing pink and there's no pink around it either. Another media which is both differential and an enriched media, meaning it's more than one category, is blood agar. Blood agar is an enriched media allowing you to grow fastidious organisms that take their nutrients from the red blood cells in the blood agar. And these organisms would not grow if you did not have the blood agar in there, meaning the blood in the agar. So it's an enriched media, and you can grow, like I said, Streptococcus pyogenes on blood agar. It will not grow on any general purpose media. A blood agar is also differential because you can differentiate the cells on their hemolytic activity. Let me blow this up a little. So right here in the white, we have in the gamma a white bacteria we streaked on the blood agar. And you can notice the red blood cells remain red all the way up to the bacteria. And that's because this bacteria has gamma hemolytic activity or gamma hemolysis. And that means that the bacteria does not lyse the red blood cells. On the other hand, with this bacteria, which is shown in the off-white alpha, is grown there. This bacteria has alpha hemolytic activity. If we look at the blood, it's red up until a dark ring or a greenish colored ring around the alpha, the growth of the bacteria. And then if you look inside the alpha, it's also dark. And the reason why it's dark or has a greenish tint to it is because some of the red blood cells have been lysed, but not all of the red blood cells have been lysed. And this we call alpha hemolytic activity. Any question about any of that? That's a very thin, dark ring around the alpha. Like I said, it's much bigger inside the alpha. The third hemolytic activity is beta hemolysis, where the red blood cells are completely lysed. So in the white beta, we have the streak of bacteria growing, and around the beta, we have a clear zone where all of the red blood cells have been lysed. This is beta hemolysis or beta hemolytic activity, where all of the red blood cells are lysed. If you were to hold this up to a light or to the clock, whatever, you can see right through here. where there's beta hemolysis of the red blood cells. You can't see where the bacteria is or where there's blood, but you can see where there's beta hemolytic activity. Any question about any of that? Let me point out that it's very normal to see a ring of alpha hemolysis, the darkening of the blood, around the beta hemolysis. That is very normal. So there are a wide variety of different media that can be inoculated to grow bacteria. This table is listing the media. Let me shrink that just a little. The first, boy, I can't get that up. Oh, there it goes. At least I can get it down. The first is the general purpose media, including TSA or tryptic soy agar, Na or nutrient agar, or Na, nutrient agar with yeast extract. This media promotes the growth of most organisms, but it does not promote the growth of a fastidious organism. Then we have differential and enriched medium. This would be a blood agar, BAP, promoting the growth of fastidious organisms like Streptococcus pyogenes. It's also differential, allowing you to differentiate the cells on their hemolytic activity. There's CAP, and this is only an enriched general purpose media. And then there's MAC or McConkey's, EMB or eosine methylene B, hectone agar or HE. These are both selective and differential media. They are selective, all three of them, in that they can grow gram-negative bacilli. So they select for gram-negative cells, particularly gram-negative bacilli. They are also differential on their ability to ferment the sugar lactose. You can see if they grow on one of these plates, you can see by the color whether the bacteria ferment the sugar lactose or whether they're a non-fermenter, cannot ferment the sugar lactose. And the color will be dependent on which media you use. With McConkey's, it'll be pink if it can ferment the sugar lactose. With EMB, it will be metallic green or a dark color. With hectone enteric agar, I believe it'll be salmon colored if it can ferment the sugar lactose. You don't need to know the color for this portion of the lab, but I think in a later lab, we'll talk about the different color of these three. Any questions about MAC, EMB, or HE agars? Where's my mouse? All right, there's also PEA, that's phenylethyl alcohol. This is a selective media. It selects for the growth of gram-positive bacteria. It selects against the growth of gram-negatives. So if the bacteria is growing on PEA, it's likely to be a gram-positive organism. What happens is there's a phenylethyl alcohol, and the alcohol gets inside the gram-negative cells. The outer membrane doesn't like exposure to alcohol, and it prevents the gram-negative cells from growing. The last media I want to talk about is mannitol salt agar. It is both selective In differential, it is selective in that it only allows high sodium growers to grow. Mannitol salt agar is 7% sodium chloride, and that will prevent most bacteria from growing. Most bacteria will grow only in a media up to about 2% sodium chloride. So let's say 0 to 0.2%, 0 to 2% sodium chloride. And beyond that, most bacteria cannot grow. Mannitol salt agar has 7% sodium chloride added to it. And so if the organism grows on mannitol salt agar, then that organism can tolerate a high level of salt. One family, there are a couple of families that will grow in the high salt, but one family is Staphylococcus. It grows well on mannitol salt agar plates. Mannitol salt agar is also differential. It allows you to distinguish between cells which ferment the sugar mannitol and those that do not. If the bacteria can ferment the sugar mannitol, they'll make the plate turn yellow. I thought I had a picture here, but I'm not seeing it. So I guess not. If the bacterial growth, especially the area around the colony, turns yellow, it's fermenting the sugar mannitol. Once again, when the sugar is fermented, it produces acids, and the acid will change the pH indicator's color to yellow. If it cannot ferment the sugar lactose, then the pH will stay about the same and the color will not be yellow. It will be, it's kind of a pinkish color, the mannitol salt agar. Any question about any of that? All right, so we inoculate the culture on an artificial medium, usually an agar plate, and usually you streak it out so you can get isolated colonies. After you're done your streaking, well, you do streak in isolated colonies to get pure cultures or potentially pure cultures. To get a pure culture, you need to pass the cells through two colony forming events. Louis Pasteur developed the concept of a pure culture, and it was very quickly learned that you have to work with a pure culture because otherwise, when the test becomes positive, you don't know which species in your cocktail of different species change the characteristic to a positive result. Louis Pasteur also developed a method for obtaining a pure culture, and his way of getting a pure culture was very laborious and took a long time. You made different dilutions of your sample, and then you plated each dilution 10 times. If one of the dilutions only had one tube with growth in it and 9 tubes with no growth in it, Louis Pasteur did correctly assume that it was a pure culture if it only grew in one of the 9 tubes. Now, Robert Koch developed a much easier way of developing a pure culture. And what he did was he streak plated the cultures on an agar dish, plating them out to get isolated colonies. And then he picked the cells from one isolated colony and streaked it out on a second media, a second plate. to get isolated colonies. And then to get his pure culture, he picked from one colony that was isolated and went through 2 colony formation events. So the chance of that being a pure culture is higher than 95%. Any question about any of that? All right, so streaking for colony isolation, what you do is you you sterilize your loop, and then you pick up the cells you want to transfer and grow on the plate. Generally, you get the cells from your stock culture. So you sterilize the loop, cool it, put it in the bacteria, and then streak the bacteria in sector 1. Then you sterilize your loop, cool it, and then streak through sector 1 to pick up cells on your loop and streak it out into sector 2 so that there will be higher cells in this region than in that region. And you really shouldn't streak all the way into streak 1. You should streak into really only this region, the lowest concentration of bacteria in sector 1. You don't want to come all the way up here because this will be the highest concentration of the bacteria. As you streak, you will deposit cells, and so you'll have more cells here. I didn't mean to do that. Than you will have here on your last streak. So you should only go about that far. You shouldn't go all the way over. And then streak out sector 2. And then you sterilize your loop. Cool it, usually by touching a sterile agar. And then streak sector 3, where you pick the cells up from sector 2. So you streak through sector 2. Once again, you shouldn't be coming all the way down here. You should only be streaking this region of the plate where the bacteria are more diluted. And then finish streaking out sector 3. Once again, sterilize your loop, cool it, usually by touching it to the sterile agar. And then this one is actually done correctly. Streak sector 4, going through a little bit of sector 3, where the bacteria growth in sector 3 are lighter, this region here, than heavy, this region here. And then streak out sector 4. The purpose of this procedure for streaking is that with each sector, you'll get more dilute bacteria, so that someplace along the line when you're streaking, you will streak down onto the agar plate an isolated cell, which will grow into an isolated colony. And it doesn't matter where the isolated colonies grow. If they grow on sector 3 instead of sector 4, that's fine. You just will have little, if any, growth on sector 4. And that's because the bacteria were more dilute than the starting culture that we did here. And it's also possible that the isolated colonies could happen in sector 2. Or even if you had a really dilute sample of bacteria to begin with, it's possible to get your isolated colonies in sector 1. Any question about any of that? All right. So incubation is putting your media, the plate or tube with the media, into an environment suitable for the bacteria to grow. Usually we call this an incubator. The incubators in Clark College, the microbiology lab, are fairly primitive. They can only control the temperature. So the incubator heats up to the temperature that you set it at. And all of our incubators are growing at either 35 degrees Celsius or 30 degrees Celsius. The incubator that we have can control the temperature, obviously the time. For time, you just put it in and then pull it up when the time is done. And the incubator can also control the humidity level. See this dish in there? Somebody put water in there, and because the incubator heats up, that evaporates the water, putting humidity into the air. This is important if you have a plate and you want to keep your plate alive for a week or more. You need to have humidity in the incubator. Otherwise, the plate will quickly dry up in, let's say, about 2 days, depending on how much media is in the plate. But after about 2 days, the either the media dries up or the media becomes so salty that the bacteria cannot grow in it. This is a fancier incubator, and besides controlling the temperature, the time, and the humidity, this one can control the levels of oxygen inside the incubator, as well as the levels of CO2. I used to remember there was a bacteria that grew better in high CO2 levels. It might be Neisseria gonorrhoeae, the bacteria that causes gonorrhea, but I'm not remembering 100% for sure. But I think that's one that will prefer growing if you add more CO2 and reduce the oxygen just a little bit. All right, any questions about incubation? If not, let's move on to inspection. Once the plate or the tube has grown, let's say overnight or after 24 hours, you pull it out and then you look to see if there's growth in the media. You can describe the appearance of colonies if the cells are on an agar plate and if they have colonies, such as the size of the colony, the shape, the margin, that's the edge of the colony, the elevation. So you can begin to get your information to begin to start to identify the species. Any question about any of that? All right. And then lastly is identification. This is when you test the isolated colonies to see what the results will be in a different test. And by running the different tests, you can identify the species of bacteria that is growing from the clinical specimen. And if it's a pathogen, you can say most likely this pathogen is causing the patient's symptoms. Signs or symptoms to be specific. All right, any question about any of that? If not, know the terms of this lab. You're not going to be performing any of these activities. Everything we do will be online, but we do have some videos for you to watch on inoculating an agar plate using the street plate culture technique. For this video, you only need to watch it for 4 minutes and 15 seconds into the video. And you should note that the author of this video only streaked out 3 sectors instead of 4 sectors, which is shown here. It doesn't matter if you only streak out 3 or 4. The purpose of the streaking is to get isolated colonies so that you can then streak it out again to get pure colonies, pure culture. Getting there. We have a video for you to watch on beta hemolysis, alpha hemolysis, and gamma hemolysis. We have a video on McConkey's auger. On McConkey's auger, you only need to watch this video for 3 minutes and 50 seconds into the video. After that time, you can cut the video. It's probably talking about something specific. And in this case, we're only looking at the characteristics of McConkey's auger and what you can use it for. And then we have a video on mannitol salt auger. So watch that video. We do not have a virtual lab for today because the virtual lab we have requires Adobe Flash Player, which no longer works. And we haven't found a substitute, obviously. All right, any questions? If not, let's go ahead and get started on the lab. Let me go to the worksheet. So for the first question, describe a natural circumstance where a staphylococcal species of bacteria might benefit from salt tolerance. So give me a natural place out in the wild where Staphylococcus can grow and tolerate the existence of salt, a fairly high salt concentration. Do not tell me something like a petri dish in the lab, because that's not a natural circumstance. And 2, how might hemolysis benefit a hemolytic organism? So here we have a bacteria that's hemolytic, and then it lyses the red blood cells. What advantage is that to the bacteria? How does that benefit the bacteria? All right, 3, imagine you have a culture containing a single bacteria species. This culture may be one of several possible species, and you don't know what species it is. Describe a scenario where you can use mannitol salt auger, blood auger, and EMB plates to help you identify the unknown bacterial species. That is, how would you use these plates to help identify the species? I'm going to warn you that each of these media has 2 properties that can be used to determine what species of bacteria the one is that you're testing. Any question about any of that? All right. Question 4. What is the main goal of streaking for colony isolation when you're streaking on an auger plate? 5. Come on, mouse. What happens to the growth on the plate if you forget to sterilize the loop in between sectors when streaking for colony isolation? In this question, you must be specific in your answer. You need to discuss the growth on each sector. So let's go to the picture here. So what you're doing is you're sterilizing your loop picking up your cells and streaking them in sector 1. Normally, you sterilize the loop and then streak through sector 1 to pick up the cells and streak out sector 2. However, in this case, you are not sterilizing the loop in between sector 1 and sector 2. You're also not sterilizing the loop in between sector 3 and sector 2, and in between sector 4 and sector 3. So explain in copious detail how the growth will look on each of the sectors. Any question about any of that? Question 5 is a thinking question. I'm going to warn you that most students get it wrong the first time they answer it. Question 6 is also a thinking question, similar to question 5. What happens to the growth on the plate if you transfer from stock culture to inoculate each sector? You need to be specific in your answer, discussing the growth on each sector. So let me go back to here. So the first sector is done normally. You are sterilizing your loop, cooling it, picking up the growth from the stock culture, and streaking out sector 1. However, you don't sterilize the loop and then streak through sector 1 to get cells for sector 2. What you're doing instead is you're sterilizing your loop, cooling it, and then picking up cells from your stock culture to streak out sector 2. Likewise, for sector 3, you're sterilizing the loop, picking up your cells from the stock culture, and then putting the stock culture in sector 3, streaking it out. And the same thing with sector 4. Any question about that? Like I said, both 5 and 6 are thinking questions, so you need to think about your answer. 7, suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. That is, you began initially with a mixed culture, but then you got isolated colonies of which at least some of them appear to be only one culture. How can you use this plate to create a new pure culture of one of the bacteria? Once again, be specific in your answer. Question 8. In the table below are a variety of agar plate media available to microbiologists. Denote each medium as non-selective, selective, or differential by placing an X in the appropriate column. You may choose more than one column. Like if you think Na is both non-selective and differential, put an X in the non-selective position and the differential. With tryptic soy agar, if you only think it's selective, put 1X in the selective media. And that's how you fill out this table. All of the characteristics for all of the media discussed were discussed in your lab module. So you should be able to get your answer from the lab module directly, at least in this quiz. In future quizzes, you will be asked a question and the answer will not be in the current lab module. You'll have to look up back log modules before you can answer the question. Like I said, you can choose more than one. You can put an X in 2 columns. In no case do you put an X in all 3 columns because non-selective and selective, you can't have a media that's both non-selective and selective. All right. For question 9, it says which agar plate was inoculated using the streak plate technique, meaning the colony isolation technique. Plate A, plate B, or plate C, and write your answer there. Any questions about this lab? All right, if there's no questions, I'm going to stay.
Dr. Volpe,
Huh?
the concept of hemolysis, am I saying that correctly?
Yes?
Okay, it's new to me, so I just want to understand, is a hemolytic organism an organism that can break down and destroy red blood cells, and therefore hemolysis is what would help that organism to grow because it's nourishing it, or am I on the right track there?
Yes.
Yes? Okay. Thank you.
All right.